Good evening, everybody, and welcome to this IF Oxford event about gene therapy for lung diseases. And here is Deborah Gill. Over to you.
Thank you, Dane, and thank you very much, everybody, for attending this evening. I'm just going to share my screen. Okay, well, yes, I'm going to talk about gene therapy for lung diseases, and perhaps some of you already heard a bit about gene therapy for heart diseases from the previous speaker. But first, we're going to kick off with a poll question. So if Dane can launch that poll, if you can respond to this question, have you or do you know anyone who has received gene therapy? So just take a few seconds for you to respond to that. So the reason I'm wanting to know a bit about this is because I'm going to ask this question again later on and see if any of your views have changed since we asked this. Okay, so everybody said no, they don't know anybody who has received gene therapy, and that's kind of what I expected. So that's great. Let's close that poll. Okay, so let's kick off with something that we do know about, and we know a lot about, at least if we may have seen it on the television, for example, is when a baby is born. So when a baby's born, you have to pause for the first breath. There's quite a lot of anxiety for the first few minutes after a baby's born, because the lungs need to inflate for the first time to take in air. And when this happens, there's a sigh of relief. And if all is well, then there's a cycle of feeding and sleeping that goes on, essentially repeat. For new parents, this might seem endless, but the baby gains weight, and that's terrific. If not, then health professionals might say that there's a failure to thrive, and this requires a diagnosis to find out what's going on. And that can be quite difficult to find out. And this is one of the ways that we find out. We do a heel prick test, which many of you, I'm sure, have heard of. So this is where we take a tiny drop of blood and we screen for up to 9 diseases in the UK. And these diseases are the ones which require an early diagnosis, because it's important to get treatment to that baby very quickly. So 2 examples of this are PKU, or phenylketonuria, where you need to have a special diet from very early on, and cystic fibrosis, which is a lung disease which develops, which I'll tell you about in a bit. In addition to the heel prick test, we can have newborn genetic screening programs, and this is rather ad hoc, and it's where we can look for genetic diseases which are essentially not screened for all children, but only when they are ill. So having a few problems with moving it. Okay, so the diagnosis can be difficult for rare diseases. So what do I mean by a rare disease? Well, this is defined as a disease which is fewer than 1 in 2,000, and that's very different from diseases such as cancer. And if you go to the Cancer Research UK website, they'll tell you that something like 1 in 2 people will develop cancer during their lives. And diabetes, that's becoming also very much more common, and in particular estimated about 1 in 10 by 2030. So those are common diseases, and that's different from what we're talking about here. I'm talking about rare diseases, and there's 7,000 rare diseases which are known. 30 million people are affected in Europe. Half of them are children which are born with the disease, and most of them are genetic in origin. And that means that they're chronic and they're a lifelong affliction, and there's very few cures. Now, things are changing with the development of charities and clinical and family networks which are emerging, and they're trying to bring together groups of people who are interested in tackling these rare diseases. And in particular, gene therapy for rare lung diseases, there's about 100 lung diseases are considered. I'm sorry, I'm having real problems with my switching here. Okay, so before I get into cystic fibrosis, I don't know how many of you have heard of the Ice Bucket Challenge. So in the summer of 2014, this was rife everywhere. This is where you had to essentially pour a bucket of ice water over your head and challenge 3 friends to do the same. And if they didn't do it in 24 hours, they basically had to give a donation to the charity for ALS. And in the UK, that's called MND, or motor neurone disease. Very rare disease, 1 in 50,000. It affects nerve cells in the brain and spinal cord, and gradually these nerves die, and the patient becomes kind of incapacitated, unable to move. Sadly, there's no cure. There's no known cause either, until the Ice Bucket Challenge, which, in addition to raising awareness, also raised some $220 million worldwide. And as a result, this was all put into research. And in 2016, a new gene was discovered, which underpinning the disease. And this has, of course, become a new target for treatment. So this just demonstrates that with increased awareness and funding, you can get research to look at these very rare diseases, because otherwise it's very tricky. And of course, on the back of this, you can develop new therapies for rare diseases. And in particular, I'm going to tell you about the gene therapy that can be used. So gene therapy for rare lung diseases, there are about 100, as I said, that are considered rare. In cystic fibrosis, this is one of the diseases that I work on. 1 in 3,000 people are affected. And surfactant deficiency, which is much, much more rare, between 1 in 100,000 and 1 in a million. So to start at the beginning, we need to talk about what actually we're trying to achieve. And to make sure we're all on the same page, I wanted to remind you that a cell is the basic unit of the body. And when I think of a cell, I think of a frog spawn. So it's a bag of jelly, which is kind of similar to the cell, where the jelly is the cytoplasm and it's bounded by a thin membrane. And in the center, the dark spot is essentially the nucleus. And it's the nucleus that contains the genetic material. Now, most cells don't look like the bag of jelly. They are very specialized for a purpose. And here's an example of a heart muscle cell, similar to the ones that the last speaker was talking about. And you can see here, it's just beating away. Even though you've taken it out of the heart, you can still see the heart muscle beating away. Here are some plant cells. These are green because they contain chloroplasts, which is what absorbs sunlight and photosynthesizes and gives the plant its green color. So these cells are very small, and we need a microscope to see them. They're not like frog spawn at all. And if we think, just to show you an idea about how small these are, this is actually looking at the surface of an oak leaf, which is of actual size. And if you use the microscope to magnify it 10 times, and then 100 times, and then right down again onto the leaf surface, and then right into the individual leaf cell, and then finally you get inside the cell to see the nucleus. So you've magnified a whole bunch just to see inside the nucleus. And then inside is the chromosome. Now, the chromosomes are just bundles of DNA, and this is a schematic. This is not a real, what they really look like. And you can see the double helix of the DNA here, but it's actually all tangled and bundled up and compressed into a small space. But what they really look like is this. There are 23 pairs of chromosomes in a human cell, and these are the chromosomes that have been taken out of a cell and they've been stained, and you can see they're all in these kind of like matching pairs. And what happens is that you inherit one of the chromosomes from your father and one from your mother, and so the child has this mixed bag of chromosomes, and this represents essentially the instruction manual for building a body. Okay, so if you open those chromosomes and you unravel all of the DNA, there's a huge amount of it, and a gene is just simply a stretch of DNA. There are some 25,000 of these genes in the human genome, and as the last speaker mentioned, the genetic code is what actually tells the body what to do to build all of the cells and all of the organs that are needed. And the genetic code is made up of these 4 base pairs. And if you wrote out all of the genetic code within the human cell, it would take some 260,000 pages. So that's a huge amount. And the most amazing thing is that when the cell divides, it has to actually replicate all of that 262,000 pages. And of course, when it does that, it makes some mistakes. Now, these mistakes are called mutations, and there are many repair mechanisms to make sure that they're not passed on to the next cell. But sometimes there's ones that escape the repair mechanisms. Most of them are benign, but some of them cause disease. Now, gene therapy aims to treat the genetic disease. Now, again, as the last speaker was talking about, ideally, what you'd like to do is just take that one single base mutation, that one single spelling mistake, and repair it completely. And that's something which is called gene editing, and that changes the genetic code. So, for example, here it would actually change this G to this T, so it corrects the mutation. Incredibly precise, sounds amazing. And again, as the last speaker highlighted, it's very futuristic at the moment. It's not yet in the clinic. Gene therapy, however, is a bit more straightforward. This doesn't worry about being very precise. What it does is it delivers a functional gene alongside the patient's own copy. So it's more straightforward, and it is in the clinic. So the greatest technological challenge in modern medicine is what's been, this has been called, and I'm going to tell you why that is. So it's all about delivery. Now, all of us are experiencing shortages about delivery, and we know how important delivery is for our petrol and our keeping our Sainsbury's supermarket shop stocked. And so what we actually need is fast, efficient delivery. So this is the gene here. We need to put it in some kind of vehicle to deliver it to the cell. Now, there's two ways of doing this. You can either use something called a non-viral approach or synthetic approach. And this is where you take the gene and you mix it with some kind of synthetic lipid or oil. And it's a bit like a salad dressing. When you mix it, you end up with the kind of oil mixing with a kind of acidic DNA, and you shake it up and it forms an emulsion and lots of tiny, tiny droplets, which you can then spray onto the cells, and the cells would just absorb the oil, and then that's how it delivers the gene. Or you can use viral gene therapy, which is where you take a modified virus. So this is a virus where you've already taken out all of the disease-causing genes in the virus, and instead you insert your gene of interest that you want to deliver to your cells. And again, this is being used in many different settings. And in particular, if you've received the AstraZeneca virus for COVID, then you will have received viral gene therapy. And if you have received the Pfizer vaccine for COVID, you will have received the non-viral gene therapy. So I just want to launch the poll again, just to check. Now that you know what gene therapy is, do you know anyone who has received gene therapy? So Dane, could we launch that poll again? Okay, have we got any results? Okay, well, that's interesting, isn't it? 100% of you now know someone, perhaps even yourself, who has received gene therapy. Okay, thank you for that. We'll close the poll. Okay, so moving on, I want to tell you about gene therapy for cystic fibrosis. And what we're doing is researching both viral and non-viral approaches. So the two types that I've just told you about. And the story for cystic fibrosis started in 1989 when the gene was first identified. And the mistakes or the mutations in this gene lead to cystic fibrosis. So it's very rare, as I mentioned, 1 in 3,000 live births with 100,000 individuals affected worldwide. And some 1 in 25 carriers in the UK. So 1 in 25 people have the mutation. So it's pretty common as a carrier, but you wouldn't know that you were a carrier because they're usually healthy and don't show any symptoms. But if 2 carriers have a child, then you have a 1 in 4 chance of having a child with CF. So this is called autosomal recessive inheritance, a classic form of inheritance of these kinds of genes. And what are the symptoms? Well, it affects all of these organs, and it mainly affects the respiratory and the gastrointestinal systems. Those are the most problematic. And if it has a very high treatment burden, so 1 month's supply of medications, this is a huge amount of medications to deliver, taking several hours a day, certainly interferes with going to school and living a normal life. When you just like to get up to go to work in the morning, you have to spend several hours delivering antibiotics, for example. But it's the lungs which are the major cause of the problems. These tend to accumulate a kind of a sticky mucus, and this causes like a chronic infection. And you get this cycle of infection that you can't get rid of no matter how many antibiotics you use, and then you get inflammation, and then you get lung deterioration. And the lung deterioration is the real problem with this disease, and it causes ultimately lung failure. This is to remind me to tell you about the structure of the lungs. So, so here you have the windpipe or the trachea, and this is where the air goes down, and it divides into these smaller and smaller airways and takes the air right to the deep into the lungs where it's needed, so that the oxygen can be passed into the bloodstream. If we take a closer look at the normal airway, so if we take a cross section of the airway here, you can see that there's this nice big empty space. We call this the lumen of the airway, the center of the pipe, and the air can travel really nicely and clearly through these airways. And this points to the target cells that we need to be able to deliver our gene to with our gene therapy. If you look at the CF airway, the same sort of cut through the airway, you can see that there's no nice clear lumen for the air to travel. Instead, it's blocked with mucus and bacteria. So what does this CFTR gene do? Well, it encodes a protein that sits in the cell membrane, and it controls the movement of chloride, so that's salt, and water across the surface onto the surface of the lung. But in the case of the CF airway, the CFTR protein is defective, it doesn't work properly, you can't get enough water onto the surface of the lung, and you get this buildup of sticky mucus, which is very dehydrated. So in 2001, I formed the UK Cystic Fibrosis Gene Therapy Consortium, along with group members from Imperial College in London and also the University of Edinburgh. And we joined together to make it our joint aim to develop a gene therapy for cystic fibrosis lung disease. And this just shows some of the organizations and some of the charities that raised money for us to allow us to do these kinds of studies, very similar to what the last speaker was talking about, and similar to the Ice Bucket Challenge. And our approach was to take the best viral and the best non-viral delivery methods. And we wanted to build on early proof of principle clinical studies. There have been some, I think, 25 different studies around the world performed to find something what we call proof of principle, which means, can we see, is it likely to work? We weren't actually looking to see whether it actually was going to work. It was going to say, is there proof of principle that it's worth taking forward? It's the first kind of trial that you can do. But our trial, we really wanted to understand, was it working? And to do that, we had to undertake a number of studies. First of all, we had to find out if we could tackle manufacturing to make sure we could make enough of our gene therapy to give it to patients. We had to perform toxicology studies. That's to find out if it's safe enough to give to our patients. And then we had to undertake our clinical trials to make sure we could actually give it to patients and find out if it has any benefit. So those are the stages that we went through. And I'm not going to go into those. I'm just going to go further forward. So we started off with our first non-viral formulation. It's the best one of about 20 or 30 that we screened. It has a terrible name, which I'm not going to read out, but basically it's a circular piece of DNA containing the gene called a plasmid, and we mix that with some liposomes or lipids in a kind of like a salad dressing approach. And we take that salad dressing and we use a nebulizer, and this delivers an aerosol, a kind of a spray of our gene therapy deep down into the lungs where it's needed. And our clinical program was in two parts. The first one was to say, is it safe? Can we deliver a single dose, a single nebulizer to patients? And I'm pleased to say that there were no safety issues identified. Our second part of the program was to give what we call multiple dose. So this was one dose per month for 12 months. And this essentially, so they ended up having 12 doses. And we wanted to know, is this effective? So our clinical trial was in about 130 patients, all over the age of 12. That was the youngest that we were allowed to treat for 12 months. Lots of visits. Patients came from about 18 different UK centres, travelling to London or Edinburgh for their monthly dosing. And this was a placebo-controlled trial. So half the patients received the drug, the gene therapy, and half the patients received placebo or salty water. And we dosed a very small amount, about a teaspoon, every month in the spray, in the nebulizer, to the lungs. And we asked the question, does the gene therapy have clinical benefit? And to find out that, we had to measure something called lung function, in particular something called FEV1, or forced expiratory volume, in 1 second. And this is a bit like if you're blowing into a balloon, you're using as much force as possible to blow up the balloon, and there's a calculation that can be done to work out how much capacity you have in your lungs. This is your lung capacity, and this goes up and down depending on your fitness in CF. And this is the how we measure FEV1. We measured it once before the 12 doses and after the 12 doses. We measured it in between, but we were only looking at the difference between before and after. And this is the change in FEV1. So we start at 0, where there's no change, where all the patients started. And then you take measurements and you find out what happens to the effect on lung function at the end of the program. And these are the results from the placebo group in red. So started at 0, their starting position. And you can see that gradually, over the course of the 12 to 14 months, their lung function deteriorated. And this is kind of what you expect for a CF patient over a number of years. So over the period of 14 months, their lung function declined. These are the results from the active group in blue, and you can see they started in the same place, but in this case, the active group showed a stabilization of the lung function compared to placebo. Now, these were, we were very excited about these results because this was the first time that any clinical benefit had ever been shown for cystic fibrosis gene therapy. As I mentioned before, there were lots of other trials which had shown no benefit, and so we were all worried that this was never going to work. But this was great. We actually saw clinical benefit. It stabilized the declining lung function in CF patients. It was also feasible for all mutations. So we had patients with all sorts of different mutations coming to us for this trial. And this is really important because gene therapy works for all mutations, and there are some 2,000 or so far identified for cystic fibrosis. And this is very much unlike gene editing, where you would have to know the exact mutation and then make the precise change. But we needed more efficiency. It just wasn't quite enough. We could have gone on and developed our non-viral approach further. We could have dosed more. So we could have given them 10 mils or 15 mils of the treatment, or we could dose more often. So we could have dosed weekly, for example, or we could have even dosed younger patients and protected them from having this decline. But remember, I also said that we were looking at a viral approach, and we took our best viral approach, which turned out to be something called modified lentivirus. And so we decided to proceed with this. Now, a lentivirus, I'm sure you all have heard of a lentivirus. HIV, that's a lentivirus, which causes AIDS. We used a kind of a sister virus called simian. So it's from monkeys, and it has no known pathology. Now, this kind of virus and this kind of approach has been successful for many other diseases. Hundreds and hundreds of patients have been dosed with HIV modified virus for gene therapy. And the advantage is that it gives very long-lived gene expression. So instead of having to dose every week or every month, we could dose maybe once every few years, which is really good. But the problem for us at the time was that it does not infect lung cells. So we had to do something really clever to retarget that virus to make sure that it could deliver the genes to the lungs. This is something called pseudotyping. And what it means is that we removed all of the coat proteins, which are responsible for cell entry from our SIV. And instead, we put a new set of coat proteins from another virus, which infects lungs really efficiently. And so this acts like a key, and these proteins can be inserted into the lung cell, and it unlocks them so that the gene can be delivered. And this hybrid virus that we've now generated has advantages both of the lentivirus, because it gives very long-lived gene expression, and also very efficient airway targeting. So it's kind of like the best of both worlds. And when we tested this new viral approach in the lungs, this is actually in mouse lungs, where we've delivered the gene for green fluorescent protein. So each of these cells here, which are very similar to these cells, our target cells in the airway, each one of these is now making green fluorescent protein. And this shows us that how easily it is for us now to deliver our genes to the lung. And it's very much more efficient than our best non-viral approach, probably about 10 to 100 times more efficient. So we were delighted when we attracted a pharmaceutical partner. So if you involve industry, then you don't have to rely on charity anymore, and you can take forward your viral approach to something you can give to patients in a large trial, for example. And we were fortunate to work with Boehringer Ingelheim from Germany and Oxford Biomedica from Oxford. And we're now collaborating to develop this gene therapy for cystic fibrosis, which I'm really excited about. However, in 2019, we decided to rebrand ourselves as the Respiratory Gene Therapy Consortium. So to move on from CF, we'd spent a long time developing this efficient viral delivery system, and we wanted to see, could it be used to treat other lung diseases? And one of the diseases that we're focusing on now is respiratory distress in babies. So at the beginning of the talk, I mentioned how difficult the first breath can be. And in some cases, that's so difficult that the babies end up in respiratory distress. Now, when a baby is premature, because this is quite common in premature babies, you can treat them with steroids to mature their lungs, because that's one of the reasons that they haven't quite got enough force to kind of like inflate their lungs properly, just like blowing up a balloon. You can give them supplemental oxygen to make sure they've got enough, and you can also put them on a ventilator. And after a few days or weeks, they usually can, or even sometimes a few months, they can actually come off the ventilator, and then they're usually fine when their lungs are mature. Unfortunately, there are some babies, particularly full-term babies who have completely mature lungs, they still cannot inflate their lungs. And that's because they have a rare genetic disease called surfactant deficiency. And it is very rare, as you can see here. And these babies, however much you treat them, you just can't get them off the ventilator. They just can't breathe on their own. And unfortunately, there's no other treatment except a future lung transplant, which is rarely happens in the UK. And this is due to the fact that surfactant, a slippery sort of substance which lines the inside of the lungs and helps keep them open, this is missing. And so the baby can't easily open its lungs and keep them breathing. And even if you make artificial surfactant, still that doesn't work. You have to keep giving to them all the time, and it doesn't work. So our program, our new research program that we are working on right now, is to develop a gene therapy for these babies. We're going to use our viral gene delivery system, and we're going to deliver the genes this time, rather than in a nebulizer to the whole lung, we're going to deliver it down the tube that the baby is ventilated with. So it goes straight down into the lungs where it's needed. And once those genes for surfactant are delivered, they'll produce surfactant normally. And we expect that once the baby has got over this initial hurdle of not having enough surfactant in their lungs, and they eventually can be weaned off the ventilator, and we expect them to develop completely normally because the surfactant genes aren't required anywhere else. They won't have any developmental issues, for example. Now, the take-home message that I want to leave you with is that these new technologies take time to mature. So we heard a bit about gene therapy for heart earlier. If you think about the first heart transplant patient, they only survived for 18 days. That was back in 1967. And today, thousands of heart transplants take place every year around the world. And this is all about public perception. So I've just drawn this graph here about this is public perception, and this is how mature a technology can become. So in the beginning, when you have a new idea, people say, wow, that's science fiction, that's never going to happen. And then when we do actually have a breakthrough, people say, wow, that's amazing, that's going to revolutionize our lives. And of course, that's very hyped up. What actually happens then is that the technology sometimes works and sometimes doesn't work, and then people say that's not going to work anymore, and then eventually it becomes mainstream. And so what happens between moving from hype to hope is a lot of hard work. And this is my amazing team of researchers who really do all of the hard work, and have been also involved in the Explorer Zone this weekend, which was really great fun. And I just wanted to leave you with a final thought, which is gene therapy fact or fiction? Lots of people that I've spoken to in the public say, oh, I've never heard of it. Gene therapy is just an idea, and it does need hard work. It's also prohibitively expensive, which is, I think, why we need to prioritize it for specific diseases, or if no other treatments are available. Some people say, oh, it's too technically challenging, it's never going to work. And it's true that it's not yet perfected for lung diseases, but it is actually already in the clinic for some diseases. So things like haemophilia. 10 years ago, the first patients were treated, and they're still doing well on their gene therapy. For blindness, you can actually hugely improve and maintain sight in some kinds of blindness these days. Gene therapy is available for immune diseases, so babies born that can't get a bone marrow transplant and they can't survive normal infections. They've been treated for about the last 15 years. There's new ones which go to the neuron, which has just been licensed in the UK for spinal muscular atrophy, and also some childhood leukemias. So, for example, if you can't get if conventional treatments have failed, you can now give gene therapy to treat childhood leukemias, and that's something which has been licensed in the UK. And of course, all of these companies are now getting more and more interested. Now they can see that gene therapy can work and that they can make cures for patients. And with that, I think I'll stop, and thank you very much.
Thanks, Deborah. Great overview of the current state of the art and some hopes for the future. It's such a long journey, all of these medical advances. And so if people have any questions, please drop them in the Q&A box. We do have one just now, which is asking, so in the context of cystic fibrosis, is it possible that the gene therapies that you've been working on, can they be adapted for other conditions, for instance, lung fibrosis, for instance, do you think?
Absolutely, that's a perfect question. So one of the reasons that we're involved particularly in surfactant deficiency is because the cells that we need to deliver our genes to are a specific kind of cell called an alveolar type 2 cell. And although it's really important for something like surfactant deficiency, in fact, it might be the only time they have a cure, there are other diseases which are also affected in those cells, and by we can then easily adapt, we think. Once we've got over the hurdle of showing that we can do this in the clinic for something like surfactant deficiency, then we can adapt it for other diseases. And the kinds of diseases we're thinking about are diseases like, for example, pulmonary fibrosis. This is something that people in their kind of like 50s and 60s start to generate these kind of like this lung disease, but we can't go there because it's quite common. But if we can do it for something like a baby, where it's very clear cut, there are no other treatments for these babies. The only decision you have is how long you're going to wait for a transplant, or you turn off the ventilator, which is absolutely heartbreaking. But if you can start there and show that it can work, then you can easily adapt it for other diseases. We call this kind of delivery system a platform technology. That's the aim of it. And if you can identify a system for delivering to the heart, you can expand it for lots of heart diseases, for the lungs, lots of lung diseases, for the liver, for lots of liver diseases. And that all hard work that we've been doing over the last 2 decades really is now really taking off, and is being applied to lots of different diseases.
So once you understand almost a general purpose technology,
yes,
and how that can be implemented, you kind of cracked it, and then it's just a couple of tweaks later on to go, oh, this could be tuned to this particular condition, or we can maybe move it over into this area.
Exactly, yeah.
So there's a question, someone's asking, what stage is the surfactant deficiency work on? Is it a few years away, or is it a bit further away than that, do you think?
Yeah, so because we've already developed the delivery, and we know a lot about the genes, it's much closer than we expect. I would personally like to say that if we can get the funding for this, and we're applying for all sorts of funding from charities and companies at the moment, that I reckon that our program to get into the first patients would be about 4 years.
Okay, well.
And that's because we think that the regulators will be very predisposed to say, yes, we can really see that this is such a strong clinical unmet need that we really need to do something. So we think that they'd be very positive about that.
I suppose if there's no other solution apart from, as you say, the terrible option of just turning off the ventilator and giving up, if the only way to move forward is something that's, you know, for some people would find a genetic process a problem.
Yeah.
But the other kind of choice to make is the death of a child. It becomes a much more sort of
yes,
difficult argument to kind of fight against, I suppose, to kind of develop these genetic technologies.
Yeah.
There's another question about, I'm not sure if I'm going to pronounce this correctly, sarcoidosis, is that right? Which, so maybe if you could say what it is, and then how about any kind of ideas for where a treatment for that or a cure for that is going?
So that kind of falls under one of the future kind of applications, sarcoidosis, and it's one of those diseases where it doesn't affect babies or children, but once we understand how to deliver genes for it, then we could tackle that as well. So it comes under the umbrella of like interstitial lung diseases and pulmonary fibrosis and things like that. So future applications which we could use for that. Yeah.
Okay. And there's another question about this, and we sort of looked into the potential time frame of turning some CF treatments into real, potential cures, or at least to make something chronic, I guess. Someone's asking, how far along are the cystic fibrosis lentiviral clinical trials?
Yeah, so again, similar. The thing about the cystic fibrosis lentiviral trial is that it will be the very first for lentivirus. And so that's why it always takes, it's always much slower to do something right from scratch. So again, we would say a couple of years away, we think from being able to get permission to go into the trial. So all going well, I'd say definitely a couple of years we hope to do that. And we are at this minute going through the stages that I talked about for the non-viral approach. So we are manufacturing it and we're doing the toxicology studies right now. So.
And just to sort of set it in context, a clinical trial for a new technology or a new drug even, we've obviously been exposed to this coronavirus clinical trial and implementation and global rollout in an extraordinarily fast and short period of time, and obviously that's a special case, and billions of people affected and huge amounts at stake if people were becoming infected and dying. I suppose these rare diseases, because there are fewer people and you're relying on much more smaller funding streams, charitable kind of organizations, rather than multinational large governmental economies kind of funding getting the kind of COVID stuff out. These clinical trials for rare diseases, it just takes a lot more kind of resource to kind of get it moving, I suppose. So there's a question around, is it likely to have something for under 12 years, so people under 12 years old having kind of clinical trials to kind of move things towards child treatments? Is there any movement in that direction?
Yeah, I mean, it's becoming increasingly more important to when you go to the regulators to say, this is what I want to do for this particular gene therapy, this particular application, you have to also include a plan for how you will deliver that to children. So it's kind of like part of the way of ensuring that whatever you're doing eventually gets to those who need it. So absolutely, you have to include that as well, and that's what you must do for all your diseases now.
And are the different procedures? There's obviously a child's body isn't just a smaller version of an adult's body. What are the significant differences between sort of how I guess interventions or even kind of diseases themselves manifest in young people and in adults?
So the most important thing I think you have to think about is that children, babies in particular, but children have all sorts of developmental issues so they have to, so for example, in the lungs, if you were delivering to an adult, their lungs are already the size that they're always going to be. They're never going to get any bigger. So when you give them the gene therapy, you give them enough to treat the whole of the lung. When you have a baby, you can't give them too much, but you have to give them enough. for their lungs to grow. And we don't yet know whether there'll be enough gene therapy for the growing lung. You have to be very careful about it. One of the good things about the lentivirus that we use is it actually it lasts a long time, so it integrates into the DNA, and so every time the DNA is replicated, the gene therapy lentivirus is also replicated, which means it will last for a long time. You don't have to dose it so often. So it's about how quickly that lung grows. Will it outgrow our gene therapy? You know, we just don't know. How fast will it grow? But I think that's the main thing, really, is making sure that you're not going to affect the development of the child.
Okay, so how many treatments would you say an adult might have, and how many treatments might a child have to have an equivalent sort of remediation of the problem?
Gosh, I think I would need a crystal ball to do that. So one of the things we will learn from our first lentiviral trial is how long it lasts. Because until now, nobody's done these kinds of studies, and we can model artificially, and we can grow some cells in a dish, and we can look at some mouse studies, but in the end, the only way we'll find out is by putting it into the patient. What would be fantastic is if we could just give them one dose and that's it. That would be amazing. We've done some experiments in mice and showed that one dose, for example, with the green fluorescent protein I showed, lasts for the whole lifetime of the animal. So it keeps on going for the whole lifetime, whole 2 years of the mouse lifetime. Now, wow, that would be amazing if we could do something like that in people. I don't think, everything from my experience tells me that that's probably not going to be the case. And we'll probably have, if we could get away with once a year for, or once every few years, that would be amazing too. So that's what I'm hoping for.
And I suppose, something that people may be more familiar with, something like cancer treatment, and it might be that if you're unfortunate enough to have recurring cancers, you go in for a regular check, and it makes the disease chronic, possibly. So I suppose that's one option, is just to prevent people from having massively kind of shortened lifespans.
Yeah.
So even if there's an annual treatment, it's better again than the alternative, which is unfortunately death, isn't it? So it's sort of any win in these things are hard fought and very much worthwhile, and there's a bit of hope there, I think. So that's amazing.
I mean, absolutely. I mean, although we didn't take our non-viral program forward, it was amazing that if you spoke to some of the people on the trial that received the gene therapy, they were just over the moon with the little level of improvement that they had from the trial. So even though it wasn't enough to take forward and we had to get a more efficient gene therapy, it was amazing to actually see that they had clinical benefit. And they said, wow, there was one person who said that she was like 14, and she said that she always used to go to the park with her brother and her mum in a wheelchair. And she said after she'd been on the gene therapy, she couldn't believe that she could just walk to the park. So, and it's a little thing like that when you think, wow, that's a huge change for somebody's life to be able to do that. So yeah.
You say it's a little thing, but that's massive.
Yeah.
To just have quality of life that otherwise wouldn't be there. And it's, and you've mentioned like where some of these kind of research techniques have been, so using kind of mouse models or computer modeling, and eventually when it gets to a certain stage, it's time to do the human clinical trials. And that's, there's a lot of kind of preparation that gets to that point. So even when you're thinking about some of these new trials that you're talking about in sort of 3, 4, 5 years, it's obviously been going on for ages to get to this point, and but hopefully, even if it's these small wins of being able to go to the park on your own 2 feet and enjoying a hug from loved ones, that's just a massive, massive
I think when I said it was a little thing, I didn't mean to downplay it. You're absolutely right. But when we report our clinical trials, you don't report this person slept better, this person didn't have such a bad cough, this person could go and play with her friends. Those aren't what we measure. What we measure is like hard sort of like facts which are approved as endpoints to clinical trial, and that does make it different, make it difficult in some cases not to capture the improvements that you can see. Yeah.
And that's, I suppose, where the regulators, people are looking for certain outcomes of medical procedures, but then on the very small scale, the human, the family, the friendship groups, the measurements there are just totally different, aren't they? It's sort of quality of life and an extra smile, in contrast with large scale population kind of epidemiological kind of numbers.
Exactly.
And that's the challenges that you've got to kind of push against as a medical researcher. And that's one of the, I guess, frustrations of being in medical research, but also one of the huge things that's so that feels so worthwhile is that you're taking the science forward, but you're also changing lives in a really significant and profound way for people. I think that's a wonderful talk. Thanks so much, Deborah, for being involved and sharing your work and continuing these excellent. Oh, yeah.
Sorry, there's one other question in the Q&A which I wanted to answer. It was about why is gene therapy so expensive? I didn't say it in my talk, but it's actually a very important question. And the reason it's so expensive is because is how much you have to manufacture and how difficult it is to manufacture. And amazingly, it is actually one of the benefits of COVID that all of the vaccines that have been manufactured, they've had to build huge capacity to make these modified viruses and these modified synthetic. And I think that the gene therapy field will benefit from that because the cost of goods will come down. So I think that that's, for me, that's actually a bonus of COVID.
Yeah, that sounds, I can believe that. And of course, there are some new vaccine centres, manufacturing facilities being built around Oxford. So if the research and the development and the manufacturing of these things all happening close together, it just, again, it's that density of medical research that all the people are kind of interacting and it's easy to collaborate. Some of the companies that you mentioned are Oxford based, which is just convenient. And there's that ecosystem. So we're really lucky to have that density of clinical work in this city. So on that positive note, so even COVID can have a positive outcome.
Thanks very much. And thanks for everybody for listening. Thank you.
See you next time.
Bye-bye.
Bye-bye.